Okay, so we'll get started. So it's going to be hands-on primarily, and if there is time remaining, and I think there will be, we'll start with the RNA-seq version. So if you don't have a laptop, I would strongly recommend that you sit next to somebody who has, so that even if you cannot do it yourself, you at least are able to get a flavor of what exactly you're doing. Okay? So this time, I would encourage you to go to this notebook and start running each step one by one. I will run some of the steps here as we go along. But it'll be easier and faster if you do it step by step rather than me showing each of these steps on the screen. Okay. So what exactly I did so far, I went to the website. So you can go to DH607 website, syllabus.html, go to syllabus. And on syllabus, you can go to hands-on too. So this is 23rd August. There's a link for hands-on 2. If you click on that link, it will take you to the notebook that I'm talking about. Everyone on the notebook? So if you don't have a laptop, you can just sit next to somebody who has, and you can do this together. Everyone here? So let's just give a couple of minutes for people to settle down, and then we'll start. Okay, everyone is on the notebook? Not yet. Go to the website, DH607. It's if you go on Piazza or anywhere, I think the URL is pretty straightforward. Just you can also search DH607 IITB. It's basically my first last name.me/DH607 syllabus. Click on the syllabus here. And there's a link for hands-on tutorial 2 session on today's date, next to today's date. That's what we are accessing. So I'll just wait for 1 or 2 more minutes so that everyone is on the same page. Then we'll start. The people who have just joined in now, we are going to do a hands-on. Primarily, you will be doing it and you'll be telling me what results you see. I have no idea what it looks like. Maybe I have some idea what it looks like, but I don't have a full idea. You're going to tell me what it looks like. So just to make sure, you go to the course website, DH607, go to today's date, and there's a link here for hands-on session 2. Can I get started? Sabyasachi, can I get started? Okay. Are you on the same page? Do you have a laptop? Okay. So we are doing a hands-on. So if you can just open this notebook. So in a different world or in an unideal world, I would have to ask you to install the variants of softwares, packages, check out whether you're using Linux, Windows, and whatsoever and what not. But thanks to Google, this is a very, very good platform, which makes it which sort of removes that barrier of being dependent on a particular software package or being dependent on a particular OS. all of you ideally should be able to get through all towards the end of this code without hopefully running into any issues. So it's should be reproducible. It should be something that all of you will be able to do. And I believe there'll be something that all of you will learn. So it took me a while to actually really think about what do I want you to see. And the answer was in the slides that I initially started. So if you remember, we started talking about sickle cell disease, right? And I mentioned that there's a mutation that happens on the HBB gene. We are going to just visualize what that mutation really looks like, okay? And I'm not going to do it, you're going to do it. So you'll be able to see the mutation that I initially showed you, does it actually happen in patients who have sickle cell disease, okay? So I'll just give you a very brief review of how I found this data set. So I was looking for data sets which are coming from this technology called whole genome sequencing. Nothing very fancy, it's basically DNA sequencing. You're doing it for the whole genome, so that's why it's called WGS, or whole genome sequencing. And I'll just show you in a bit how I found this data set, or how can you find data sets that you will find of interest going forward. Whether you have a laptop. So we're doing a hands-on. So if you can go to the course website. Okay, so hopefully it took a while for us to settle down, but we can get started now. Everyone has either access to a laptop or a friend's laptop. Okay. So go to the course website, open this notebook. Okay. So there's some steps which will require some time, and there's some steps which will, you'll have to restart your sessions and so on. And I haven't been able to fix it, but I think that it's fine. So the first thing that we're going to do is we're going to install a package manager called Conda onto Colab. The reason we are doing this is because we want to install some of software packages. An ideal way to sort of install these software packages would be that you compile them from scratch, but that will take time, and you have to download gazillions of dependencies, and we don't want to do that. So the first step we do is install Conda. Okay. So if you go to the notebook, the first session or the first cell just installs Conda. It will automatically restart also your session. So I'll do the first two steps, and then let you and then I'll let you wander in this notebook and figure out what do you see in the end. Okay. Everyone with me so far? So we'll hopefully not run into network issues. The other reason to choose this was the Collab servers sit on the US side. At least some of these servers are hosted on the US side. And the data set that we're going to download is also hosted in the US servers. So downloading on Collab is faster than downloading on the IITB network. Does that make sense? Okay, so we have restarted our kernel. So people who have just walked in, we are doing a hands-on session. We are working with a Colab notebook. If you go onto the course website and go to 23rd August row of that table on the syllabus, you'll see this notebook. Okay, so my kernel would have restarted. I have probably installed Conda or Mamba. Mamba is the faster version of Conda. Okay, next thing that I'm doing is I'm installing two more packages. One is called IGV notebook, and other one is called multi-QC. And you'll see why I'm doing it very early on without actually giving you any perspective of why we are doing it. It's because, again, these have like gazillions of package dependencies, and one of the package dependency has some conflict, which asks you to restart your session. So once that step is run, just restart your session. If anyone comes up with a fix for this, you get 1 bonus point, okay, so that we don't have to restart session multiple times. So hopefully we'll just do it 2 times. Okay. So once this stuff is green, it's in the background, it takes around 51 seconds for me to run. I will just restart my session. Okay, that's the only time hopefully we'll have to restart our session. So when you restart your session, the runtime is still running. It doesn't get disconnected. So you don't have to rerun these steps, right? So this step, I mean, you can rerun it, but nothing new will happen because everything is already been installed. So I've already restarted my session. I can just check whether Mamba or Conda is installed. So when I do Mamba, so Mamba is a faster version for Conda. Conda is a package manager, which we'll use slightly later in the notebook to install a few packages that we require for analyzing this data. Okay, so Mamba is working. Okay, and then I will start my timer. This timer does not really matter so much, but the notebook will take 20 to 25 minutes for you to run from end to end. What is this notebook about? So we talked about this disease called sickle cell disease. Sickle cell disease involves a mutation, which is A to T, that happens in exon 1 of HBB. So hopefully you'll recall this version of the picture that we had seen. So people who have sickle cell disease or SCA, in both copies of their DNA, there is a mutation in the valine where the GLU changes to valine. And this is happening just because of one single mutation, A mutating to T. Okay. What we want to do is we want to see whether if we get a patient who is suffering from sickle cell disease, do we really see this kind of mutation or not? Okay. So that was the question I had in mind. And then how did I find this data set, right? It's a very interesting data set, which is talking about how do you actually cure sickle cell anemia. And one idea that had come from right side of this class at some point, and I don't recognize anymore who it came from, was to do some sort of gene editing, right? So they edit that particular mutation, and then you will have fixed this mutation from A to T. So we have actually data for that in this data set that we're going to talk about. It's slightly bulky, so we'll not be able to analyze it, but I will strongly encourage you that once you go back, try to find which data set I'm talking about, and I'll give you some preview of what I'm talking about, and see whether that editing technology, does it really fix it or not, okay? For that data set, you not have to do anything different. It just is slightly intensive. The total size of that particular data set is slightly larger, so we cannot run it on Colab, but if you download things on your laptop, you should be able to run it. Okay, so how did I find this data set, right? So I went to SRA. So you can search NCBI SRA. The URL is, I think the URL is also there in the notebook somewhere. If you go down, it says the data set is available on SRA. I click on this link. It will take me to the exact study where the data set was deposited. So this paper comes from this data set comes from this paper. So it's a 2023 paper. So as recent as it can get, you're analyzing data which is right off the press, right? So it just came out like 6 or 8 months ago, and we're now reanalyzing this data set. So this is the power of or the scale of things that we're talking about, that we can just pull out any data set that has been publicly deposited, and we can reanalyze it whether the results really hold true. What they actually report, so just to give you a bit context, it's very technical. The paper is slightly technical from the biology point of view, but I strongly encourage that if you are interested, go and read it. But I'll just give you a very quick picture of what that data set tries to do. So it says that there is a mutation which in the HBB gene, and we want to be able to fix it. Why are we trying to fix it? Well, other people have also tried to fix it, but the problem with other people trying to fix it is there are a lot of these off-target effects, or the delivery is not complete. So when you deliver a mechanism which will go and induce or reverse this mutation, then a lot of things go haywire, like other things also change. What they discover or what they invent in some fashion is a new delivery mechanism using a virus. This is called AAV, adenovirus. That helps you sort of control for all of the things that can go wrong when you do this therapy. So it's a gene therapy paper. It's a paper which proposes a therapy for treating sickle cell anemia. And if you recall, I had showed you that, or I talked about that not this therapy, but a slightly different therapy has been approved by FDA. So people who have sickle cell anemia can undergo gene editing to fix their mutations. This is not FDA approved, it's preclinical, which means it's not even made to the clinics yet, or it's not even actually went into clinical trials, but it's still promising. and it's a slightly different way of fixing things. So back to how I found this data set. So if you go to SRA, so this particular data set is the entire record of what was submitted, right? So this pulls down a lot of information about what exactly that project was about. There's an abstract and so on. What I did is I went to SRA, so ncbi.nlm.nih.gov. If you search NCBI SRA, let's do that right now. The first thing that comes is what I'm talking about. So SRA stands for people who have used this earlier. What does SRA stand for? Sequence Read Archive, right? So Sequence Read Archive, right? So Sequence Read Archive, this is basically an archive of the sequencing reads. What did I search for? And I hope I remember it right. We'll try to do it now. So I searched for, if I remember, I searched for sickle cell anemia and WGS. Not this one. Okay, yeah. So I searched for sickle cell anemia, and the first few data sets that come about to be are actually also the data set that we are analyzing, right? So you see these pics of Cast-seq. So we'll not talk about this exact technology, but this is just a specific way of capturing one particular or different particular regions of your genome. So that's what Cast-seq is. And this is actually the data that you're going to analyze, right? So you see multiple records here. These records mostly come from this data set. So you see all these Cast-seq. The thing that you're seeing here are these accession IDs. So if I click on this particular link here, it tells me a record about what this data set is about, right? So this is the paper. This is a study that this data set came from. This tells me what sample it is. So this is coming from a donor who's edited, so whose genome has been edited. And then the different runs, right? So just a very quick note about runs. So the way sequencing experiments are done is you have an experiment or you have a sample on which you will run different rounds of experiments, right? So different samples or the SRX ID that you see here somewhere. The SRX here that you see here, that's an experiment run. Excuse me. That's the experiment that was done on a particular sample. Now, this is a biological replicate. Okay? So if each SRX run that you see, that's one biological sample. Each biological sample can be sequenced multiple times, and that's what makes it a run. Okay. So one sample can have multiple runs. In this case, there is only one run, which means that this sample was sequenced only once. But sometimes what happens is you could sequence that sample on, maybe you do a shallow round of sequencing first, and then you sequence it again. So it becomes like a second run, third run, fourth run, and so on. These are the SRRs, are technical replicates. The SRX is a biological replicate. Okay? Just something that you should keep in mind. Why am I talking about this? Because at some point you will do projects, and you will have to think about what kind of projects you want to work with, how you actually analyze this data. So this is a prelude to that. So we'll do some of these exercises again and again, and this will become clear. But just keep this in mind. SRX, X stands for experiment. SRR, R stands for a run. Okay, runs are generally technical replicates. Everyone with me so far? Okay, so we have found our data set, right? How did I sort of go deeper with it? So what you're seeing here is one experiment, and each experiment will be associated with a project. So in this case, this is this entire project. So if I click on that entire project, this is the link that I get, and this has different samples. There are 94 experiments coming from 64 samples, and there are 94 runs. And there is a total of 450 gigabyte base pairs or 150 GB data. So 150 GB data right now sitting on your browser. Okay. Ideally, you could have analyzed all this on the cloud and just get done with it. But unfortunately, we are limited by what Colab gives us, which is 12 GBs of RAM. So we'll not be able to analyze the entire project. We just choose one sample that we can. Okay, everyone with me so far? Okay, let's switch back. Thing that happens in a lot of these projects is you get like these gazillions of samples, but you have, yeah. So samples, yeah. So you're talking about the sample here. Right. So these are biological samples, which means 64, let's say, slices of tissue or 64 cuts that were done in, it could have come from 64 different individuals, or it could have come from one individual, but cut 64 different individuals. So these are, in some sense, biological replicates. Okay. Experiment means you can take one sample. So good, very good question. So you can take one sample and you can do multiple experiments on it. So I can take, let's say, part of my liver, that's sample one. The first, I keep it for 2 days. I run a whole genome experiment. That's experiment one. I keep it for 5 more days and run more experiment. That's experiment two. So the runs can happen for the same experiment. So I did on day two, I did it at, let's say, I did a shallow round of sequencing with that sample. And then I repeated it again on day two with a much deeper round of sequencing. So that becomes a technical run. So that's a run. So I will explain this in a new slide. Actually, next class, I should have done that. But very, very good question. Okay, so you can start from samples. Samples are basically your tissue samples. Experiments are what exactly is the analytical question that you're asking or biological question that you're asking. And runs is like, in some sense, how did you do that experiment? Like what technology you used, how many reads you sequenced, and so on. Okay. So samples is a superset of experiments. Experiments has multiple different runs. Is that clear broadly? Okay, so the first thing that happens is like, so if you go onto this record, right, like there are multiple different samples here, like 94 different experiments. Now let's just look at that. Like, what are these experiments? So it gives me this weird looking, like, how do I actually make sense what sample is doing? Do I have to go through this entire list and see what sample is what? I could send them to I can send them to something called the run selector. So let me just go back because I thought I moved very fast. So I could click on send results to run selector. And now it will give me these tables. So I know there are 94 experiments, but I have no idea what those 94 experiments are, right? So I want to see what is it? Is these 94 experiments all associated with editing? Are these positive controls? Are these negative controls? Is something really going wrong with one of the data sets? Is it one donor? Is it multiple donors? What exactly is happening, right? And you'll bear with me, like what right now is happening. Like it's just waiting for this thing to load. And when I started working in this area, this was for me the most frustrating part, and it still remains the most frustrating part. I want to just see what data do I have access to, and it just keeps on loading, and it will take another 15 seconds more to fetch. I just want to get an idea of what data set I'm looking at, right? So here, this is the table that I'm looking for. I'm looking for what is the run, what is the age of the donor, what kind of assay we are looking at. So Amplicon is just a targeted whole genome sequencing. So we're going to work with Amplicon data, but for all practical reasons, this is whole genome sequencing data. And then you have these multiple different metadata associated with it, right? So you have which location this data came from, or which country this came from, what is the instrument, next seq. So this is an instrument that you would see on one of the slides in the previous lecture. And then you have what is the sample name, which tissue it came from, what was the treatment, and so on. And this is the column that I'm actually mostly interested in, right? So there is edited, which means that gene editing has been performed in the sample, and there is mock. Mock means control. So this particular patient had, to start with, they had a mutation in their genome, which led to them being sickle cell disease or having sickle cell disease. Everyone with me so far? So 94 different samples, and this is what I wanted to look at. What should I actually analyze, right? Like okay, there are 94 samples, but how do I download them? How do I analyze each one of them one by one? How do I actually proceed? So this is step 0 before you even start any analysis, that you look at the metadata that is associated with a particular public data set. Okay, how would you how could you like, how do I actually use this information? So I could have downloaded it, right? So there is this button which I can download, and it will give me an entire table download in this format and another 15-20 seconds to download. Okay, so let it download. I'm not going to look at it. You're welcome to look at it. I've looked at it multiple times and I don't like it. So what the first one of the first problems that I actually started to think about when I was working with a lot of these public data sets is how do we analyze the metadata itself? So I wrote a package. This is, I think, now 6-7 years old. I don't recall anymore. But what this package tries to do is I give it the project ID, the SRP, and it gives me a data frame of the entire metadata that you saw there, which you would have otherwise had to download manually and see. Okay. So I gave it just the project ID. So SRP 501596 is the project ID. And you get all the that's associated with this experiment right in your notebook here. You can write this to CSV, put this up in Excel, and do your sorting and so on. Okay. So I did not put this package just because I wrote it here, but I think it's really, really useful. And I hope people who are going to work on projects will find this useful. Just to make a sense of what exactly are you really looking at, right? So right now you're looking at 94 experiments. There are experiments on SRA, which have 1,000+ different samples. So just imagine going through that painful route of, fetching that data, waiting for that loading sign to end and somehow downloading it and so on, and then analyzing. This makes it much, much simpler. Okay. Everyone with me so far? Okay. So there are multiple different samples here, right? How do I analyze it, right? So one of the first things that I could do is I could look at, you could do, now this is data frame. So this is in your territory. You can play around with this data frame. The first thing that I would encourage you to look at is the library strategy. So this notebook is in some sense a bit filtered, but let me add one more column here. So there are different library strategies, which represents where exactly this data came from. So there is amplicon sequencing, which is whole genome sequencing. There are 50 samples. There are 30 samples of RNA-seq. There are 10 samples of other. This is actually single cell RNA-seq data. We will make this story complete one day. When we talk about single cell, we'll take the single cell data from this project and reanalyze it. Okay. We will stay with this data for a while. Okay. So what I did is I said, I am not interested in RNA-seq because I've not taught you RNA-seq yet. So let's just remove everything which is RNA-seq. So we are left, or the other way to do this would be to focus on the amplicon sequencing, which is what our focus will be today. And if you look at these filtered list of samples, then we get the experiment description tells you what exactly that sample is about, right? So we have samples of P53 exome. We'll not talk about this particular sample for this part of the class. But again, I'll encourage you to analyze and go back, reanalyze this and this data set and see what insights you get out of it, right? So P53, anyone recognizes what is P53? Tumor suppressor, right? So everybody knows it's a tumor suppressor. TP53 or P53 is a tumor suppressor. It's a protein which, if bound, will suppress the expression of the gene. In most cancers or in a lot of cancers, this binding mechanism of this particular transcription factor goes haywire, and that's why the genes start to express more, cells divide more, and that's how cancer develops. So you could connect the dots and see why that data set exists here. I will not go into the details, but you can go back and reanalyze. We'll work with Cas-seq. We'll work with Cas-seq of the mock sample. Okay, so they started with this mock sample, right? This mock is the control. So the first thing that you check in any experiment is, does the experiment even make sense? What they started with, does it actually even have the disease or not? So the goal for this workshop or this tutorial is to just tell you, or to just to show you whether this mock experiment, is it correct or not? How do we identify whether it's correct or not? Well, if there is a therapy to fix it, we should at least be able to see whether that whatever is being fixed, did it actually exist to begin with? Okay. Okay, now it's on you. So take it forward from here. I'll just keep walking, and if you have any questions, I'm happy to. And I'll keep coming back to the board and talk about the perspective of why we are doing some of these things. But some of these steps are really straightforward. The first thing that we are doing is we are just choosing a sample to begin that we'll be analyzing. So if we have to find a sample, we have to download it. And one of the advantages of this library that I'm forcing you to use is it gives you actual FASTQ links. If you have worked with this kind of data or from SRA data, you know probably some of you will know how much painful it is to get the FASTQs. NCBI likes its own formats, so it deposits all the sequencing data into this format called SRA. It's a very painful format to work with because you download the SRA format, you have to convert it to FASTQ. That takes time, and it just blows up your hard disk and so on. So ideally, it is supposed to be less bulkier than FASTQ, but I don't think it really, I mean, it kind of works, but it's not really useful if you want to do this kind of analysis. So we download directly the FASTQ files, okay? Everyone remembers what a FASTQ file was? So I'll do some switching. I'll keep on doing some switching, which is just to recall what file we are downloading. We are downloading a file which is called FASTQ file. It has 4 lines repeated multiple times. Each such line represents the read that was sequenced. The first line is the read ID. The second line is the actual read sequence. The third line is a separator plus. Fourth line is the quality scores of each of these individual bases. Okay. And now you'll analyze all of this right in front. So all of this will start to make sense in just a bit. Okay. So everyone has these links figured out? Okay, so what I'm doing now is very simple. I'm just going to download those particular files that you see here. Okay. So this is one of the FASTQ files. This is the paired end sequencing data. Everyone remembers what is the paired end sequencing? So in paired end sequencing, we don't have the marker today, but we're trying to use the props. Okay, so this is R1, this is R2, and this is your DNA insert. Goal is to sequence this. You start one from this end, you start other from this end. This is paired. And if we didn't have this, that would have been a single end sequence. So this is a paired end sequencing. You require paired end sequencing. Generally, paired end sequencing is good. It gives you more resolution to go back and map this particular insert in a much robust fashion as compared to single end sequencing. Okay, is the download complete for people? I think it takes a while actually. So this step just downloads these 2 FASTQ files. So I'm downloading R1 for a particular sample and R2 for the same sample. What's the DNA insert? So DNA insert is a insert. So you had this genome to start with, right? The second step that you did is you sheared this apart. You introduce these random breaks. So these are your fragments. And now you want to sequence these. Okay. So I take one of these fragments. Okay. I attach adapters to it. So this is your adapter 1. This is your adapter 2. Next step that would happen is, since you have this adapter, you can start to amplify the sequence. This is what happens on the flow cell of the DNA sequence. So this, once you attach the adapters, it will go sit on this flow cell. So I'm just going to call it FC, flow cell. It will get amplified here. It will make multiple copies of just this fragment. And then this portion, the fragment that you wanted to sequence, that is the insert. It's just a different name for that. It's called the insert. So your sequencing will begin, if it is R1 and R2, it will begin from this side, it will begin from that other side, and you will get an insert. This is what you ideally want to sequence. Does that make sense? So your DNA sequencer will give you the information for A, C, D, E, and so on. Now this will be, the sequence that you get is 150 nucleotides long in this case. You will see just a bit. But the fragment that you shared would have been much longer. Okay, you're limited by the technology, so the technology only sequences 150 base pairs, right? So your fragments would be this fragment 1. This is, let's say, fragment. So this is, in this case, when you make this shear, right? That's when your insert size is determined. At that step, you ideally want to make inserts which are of like similar length. So if you were to plot the histogram, I could take all these fragments and plot the histogram of their lengths. And the peak that you get, that's your insert size. Which means that this is the general distribution around which all your fragments vary around. And this depends on like what kind of shearing technology you're using. It can vary from 400, it can go very small also, it can go all the way to 200. But think about it, like if you have a technology that is able to sequence 150 nucleotides, or even actually more, you want to keep this fragment length larger than. Okay, does that make sense? But when you take this fragment, right? So when you do the sequencing, you will only get part of it. So this will be 150, let's say. This from this side is 150, and the remaining is not getting sequenced because it's much, much longer. You can have a different scenario. You can have a scenario where this is your R1. This is 150. Your fragment is actually much smaller. So it's less than, so maybe it's 70 nucleotides. This is your insert, or this is your fragment. So I will keep using these terms interchangeably. Fragment or DNA insert are the same things. So you will only see a 70 nucleotide length sequence here. But this doesn't make sense. It will likely not happen that you get a shorter read. Every time you see a FASTQ file, all the read lengths should be the same. How do you make up for it? How do you make up for the remaining 80? Actually, this is slightly blown up. Let me be a bit more reasonable. So you get 130 as your fragment length. What do you think happens to the remaining 20 nucleotides? Dummy values. Okay, so it's not exactly dummy because you have these adapters here which generally will not get sequenced. There is no reason for you to be able to if this is your adapter 1, this is your adapter 2. Why do we need it? Why do we need adapters? Yeah, so for the enzyme, for the polymerase to bind, right? So the polymerase will recognize this is a fixed sequence. It will recognize it. It will start synthesizing the DNA. This is fixed sequence, but repeated multiple times. You somehow ligate it to all the DNA fragments. This becomes your startup kit for the polymerase to come and start synthesizing the DNA in the other direction. Okay. So ideally, the sequencer is smart. It knows that this is the adapter. Why waste sequencing? Why waste, like, this is a fixed sequence. Why should I even sequence? So it would not start at the 5' end. Your polymerase starts at the 5' end. It would not sequence this particular fragment. Okay. Now, in the case where you have a 150 nucleotide, say, rather longer fragment, then you can obviously keep on sequencing and you're good, right? Then you will get like a 150, you will get a 150 nucleotide read. But in the case that the fragment truncates before 150. So this is 150 and it truncates before. What it will have to do is it will have to say, okay, I don't have any other option. Let me just go ahead and sequence this adapter as well. So this thing is your adapter. Okay, so if you think I'm the adapter is on the other side. So the adapter is actually on the both. No, so the adapter is actually on the both sides. The adapter has a complementary. The adapter has a complementary stand in terms of what you're actually sequencing. Yes. Okay. So in this case, so I'm jumping somewhat. Like I'm just trying to give you an intuition of what exactly is happening, right? So in some cases, you will start to see adapter contaminate. Your reads will also have adapter part sequence. And you'll see in this notebook that we get rid of this. Okay, you'll see a plot and it will become much more clearer what it is. So hopefully the data is downloaded for everyone. Okay, let's move on next. And what I all talked about will become very, very clear now, hopefully. So anytime you look at data, right? So before you start to process anything, like you should always visually check what you're looking at. Like a lot of, so try to be a bit more cynical here, which is like a lot of if you're working in this area of, or at least I've seen people who work in this machine learning thing that they take one data set and just try to apply all possible algorithms to it without actually looking at what is the data really telling you. Don't let the machine figure out things for you. You can be a bit smarter and you can actually help the machine. So try to like keep an eye open of what exactly you're looking at. Okay? Don't leave everything to the machine. The machine will eventually figure these things out, but if you actually help it, you'll move quicker. Okay, so that's just a side note. So we are going to look at what data exactly we are looking at. So the first thing that you can check is you can check the size of your data set. So this is very small from what you would generally get. This is around 70 megabytes and 65 megabytes. They're not of the same size. Is that okay? R1 and R2, their sizes are different. Is that okay? Should they be equal? So they are sequencing R1 and R2. R1, R2. 150 nucleotides each. I'm also telling you what is the length. Okay, and why should they then they be equal? So the data will be similar, that is correct. Is there any reason that it can be different in size? Unless R2 sample and R1 will sample. Oh, I see what you're saying. Okay. Okay. So we'll see in just a bit what is really different between the size, but it's okay for these sizes to be different. How do we check whether these things make sense? We will first count how many reads are there in your data set. So we'll have to install a utility for that, BC. You can do it without this also. Like you do not actually need to install this as such, but then you'll have to calculate in your mind how many reads this particular sample has, right? So we have around 944K reads in our sample R1, and it's the same, hopefully should be the same number in the second sample, right? So before you begin with, you're just checking total number of reads are the same in R1 and R2, okay? So you're sequencing exactly the same number of fragments in both R1 and R2. Everyone with me so far? Okay. What is the length of the reads? So I'm just looking at the first 4 records. You can do a bit more. You can do up to, let's do it up to 5. So zcat is the command. So cat is a command for concatenating. Zcat you use when you have a GZ extension. And you can look at the first 5 records. Okay. Let's do the same thing for the R2. Okay. So what you're seeing in the top here is the header of the read. This is the read, entire thing is the read ID. This is the actual sequence that we get. This is the separator, and this is the actual quality that we get. Okay. Everyone with me so far? Okay. The next thing we're going to do, and this I will, I think this you should help me figure out. I wanted to calculate the length of the read, and you're seeing here 152, and I can tell you this answer is wrong. Okay, I cannot figure out why this answer is wrong, and in this case, I actually don't know. So if you can figure out what is the right command for telling me what the length of this sequence is, that'll be wonderful. One bonus point for that using bash. Okay, I can do this. I can do this thing. Okay, so it should be 151, but I get 152. What is the right command for doing that offset of -1? If you can tell me, you get one bonus point, one absolute bonus point. Okay. You can post this on Piazza if you figure it out. I was not able to figure it out. Okay, so we have 151 length base pair sequences. The next thing that we want to do is just do very quick quality control check. So I mentioned that whenever you get data, each data point that you have, as you sequence this particular read here, you get a quality for each of these sequences, right? So it can go up, it can go down. I want you to run that and tell me what happens to the quality of the sequences that you get or reads as you move along the read. So there's a tool called FastQC. It's again slightly outdated tool now, but one of the good ones that still exist out there. I'm going to just run FastQC. FastQC stands for I'm doing a QC on on the FastQ file. It's going to analyze the data for me and tell me what the quality scores for each of these reads look like. What is the general distribution of the scores that I get? So one project idea, like you can see how slow this is, right? One project idea would be, how do you make a tool like this much, much faster? It's slow, and if you go and see the code, you'll figure out why it's slow. Just written like 15 years ago. There'll be no updates about it. If you are able to come up with it, like maybe create a clone kind of this tool for analyzing this, that'll be wonderful. Okay, so while you're doing all this, right, what is happening on your files on the, if you open the file column here, is it has generated, FastQC has generated a bunch of files onto your here. So there is fastqc.html and there's a fastqc.zip. You can download it and you'll be able to see what it looks like. So I'll do it right now. Okay, so what you're looking at right now is on this particular histogram, you're looking at what is the position of your read, what is the average quality of the individual bases that comes out of the sequence. Okay, what is the trend here? What trend do you see in terms of quality scores? So it decreases with the read length, or it decreases as you move along the read. Any ideas of why the quality should go down as you go longer or deeper in your read? Pooja? I'm just looking at one read, just the forward read. So think it from a technology point of view. Okay, and why? Why is that? So I just revise the principle on which this is based, like very, very quickly. So you are on this is your flow cell FC. You have this fragment, which is being sequenced. So you'll go back to this slide and revise it, but this I'm just making a visual thing here. So the first thing that will happen is, let's say the sequence is A, C, E, and G. This will get complemented. A polymerase will come. It will deposit a base, which will be T here. And this is fluorophore, right? So this emits a light every time the incorporation happens. And then when the incorporation happens, there is a camera sitting at the top, which is going to record what does the sequence look like. It will record a T. And then it's going to wait there. Why is it waiting there? Why is the camera going to wait there? Or why is, once you get one sequence, why do you have to wait? This we discussed in the class. Right. What's your name? Oh, you can see. Okay. You requested a Google counter. So you have to, there's a fluorophore, and that was the blocking group, right? So without drawing everything, I'm just taking a brief detour. So there is a blocking group here in your chain, which was the fluorophore sequence. If there is a fluorophore which is attached here, it cannot be further extended. So what happens is you get rid of this fluorophore. You have to wait till that happens. Then you come and attach an OH group so that this can now continue. At this step, polymerase is waiting, your camera is waiting, everything is waiting. The unfortunate thing with polymerase is it doesn't really like to wait. It just wants to keep going at one point. And at some point, it will run out of gas, literally. That's why as you move along, this here, it becomes harder and harder for the camera to test or to tell very clearly what base did I see. There is fidelity. So there's a technological problem, both biological and technological problem that we have. Okay, so that's why the sequence quality drops as you move along the read. Is this clear? Is this idea of the sequencing part clear? I want that to be super clear. It is very, very important that you understand how exactly you're getting this data. Okay. How does sequencing happen? That is clear, right? You start from a flow cell. There is a sequence that is tethered to it. This is the read fragment you want to sequence. The polymerase will start. It will incorporate one base at a time. That base has a problem that, not a problem, it's a bug. It's a feature, but not a bug. The feature is that it emits a fluorophore. But because of that feature, it cannot extend anymore. So something has to come, wash this fluorophore off before it starts again. And all this time the polymerase is working. Okay. So we'll move on to what we were doing. You can then run something called multi-QC. This is slightly fancier version of concatenating. So right now when I run fast QC, I ran it on individual files, but I would like to see in entirety, if I have like 1,000 samples, what is the quality distribution of that? Yes. Good, very good question. Yeah, very good question. So this is happening at the cluster level, right? So So this is your flow cell, and there is a cluster of sequences sitting here. That's what you say, right? So this is cluster 1, this is cluster 2, and so on. The camera, this is actually in sync. Okay. So this base pair extension that you see here will be associated with some other base pair. Obviously, it will not be the same, but this is actually happening at the cluster level. Yes. Okay. But those reads are in sync. So you should still be able to, and actually that's why you're doing this cluster sampling. So all these reads are the same, right? So read, this has been amplified sitting on that flow cell. That's why the bridge amplification is important, right? So what happens is this is what your flow cell begins with. It will do a bend. It will bend over, make multiple copies of it. It will create a bridge, and then you will fold it back. This is amplified reads. This is PCR on the spot. Okay? That's the innovation that I was focusing so much on. That I almost got emotional. Like, how do you figure this out? So this is that innovation. You're doing this at the flow cell at the spot level. multiple copies of the read sitting, because if you don't have this copy, your camera will have to be extra sensitive to detect what fluorophore was in it. But this solves the problem of cluster ambiguity. They're happening in all the reads. Yes, they are happening in all the reads. Yeah. There is abundance of nucleotides and there is abundance of polymerase, so it's happening in parallel everywhere. Okay, very good question. Does everyone understand what is happening? Anyone not clear what I just draw or what I just drew? Clear? Okay. So there is some discussion. So if you look at if you download this multi-QC report, and you can do this without anything, you will see it looks like a fancy version of things. There is a sample here. It is the number of duplicates, and you will see there is a problem here in terms of the duplicates. This is amplicon sequencing, so you can trust me, you can just ignore this part. Ideally, what you want to see is, are there a lot of duplicate sequences in your data or not? And generally, this number should be very low. This is the percent of GC content. This is 40 to 50%. For this particular sample, ignore it. I'll not go into the details, but for now, you can just ignore these high number of duplicates that you see. Okay. It also tells you about which of these reads are unique in a much more granular fashion. Then it tells you about the same thing that we saw earlier. How does the mean quality look like? And it decreases as you move along the base. And your average quality scores per base are around in the 30 category. So if you remember in the slides, I had talked about how do you interpret these quality scores. So a quality score of 30 means there is a 1 in 1,000 chance of having committed an error, which is very low. Okay? 20 means 1 in 100, 10 means 1 in 10. Okay. Okay, so all good so far. Now what we're going to do is we're going to remove the adapters. Okay. So there is some discussion here, and you're welcome to read it, but I already gave you the intuition of what happens if you have. Sometimes you will see an adapter sequence, and sometimes you will not see an adapter sequence. This depends on what is the fragment length that you're seeing. Okay. So when I started doing this, right, like it was 9 years ago now, I was like, there are like a lot of these workflows. So you look at these workflows and you just like blindly copy it and like without actually understanding what do they do. But this is what they do actually. So it took me a while to figure that out. Like, why are we doing this? Because this not there in any textbook. This was not there in papers. Some of it is assumed that you will understand. But this is the general idea. This, by the way, is taken from Illumina's website, right? So Illumina is the company that does it, which kind of explains it, but you will not find it generally. It is taken as a thing that you just should understand. So we have to get rid of the adapters. The adapters are these fixed sequences that we don't want to analyze. Is it okay if I keep the adapters? Like what could go wrong if I were to keep the adapters? The sequence in what sense? Because the adapters are very important. So adapters are fixed sequences. They are chosen so that they are very unique, which none of the genomes ideally should have. Okay? It's that unique of a sequence. It's not exactly technically that unique yet, but I mean, you can look at it like what is the probability of a 20-mer finding a match in a 3 to the power 10 billion base pair. So there is some probability which is definitive, but it is designed to be unique enough so that you can cut it out. You don't want to retain it because when you start to do the mapping, this will negatively impact how many number of instances you see. Okay, so you don't want this ideally. So we're going to remove it. And this is another tool called FastP, which is actually very fast, and it removes the adapters and does some quality trimming for you also, right? So I keep quality scores which are less than 10 in your data because you're not really confident, so you just chuck them out. And you can do this easily because they're anyway going to be towards the end of your sequences. Okay. So FastP will run hopefully quick. For people who have actually proceeded forward, you can move faster than me. You don't have to follow me. The next thing that we're doing is the key element that we want to talk about, which is we're going to do the alignment step. Okay. So everything is complete so far. We have the data. We have cleaned it. We've removed the adapters. We've removed low quality scores, indexes of it. Time is now to do the alignment. And this is where the previous lecture comes into picture. So you have these reads, like now you know this is around 1,000,000 reads that you have. You want to map these 1,000,000 reads onto your genome. Ideally, what you would have done is you would have downloaded the entire genome sequence. Okay? So this website, UCSC, is one way to download it. You can go to this particular website. You can download the entire human genome in a FASTA or a FASTA file. This is that location. We'll not, unfortunately, be able to map it to the entire genome because, again, it will A, take time, and B, it blows up the memory to some extent. Not a lot, but some extent. So what I want to do now is open your, you can keep a tab on your resources column. Okay. I am going to unzip it. So I downloaded the chromosome 11. So HBB, that gene that we are interested in, sits on chromosome 11. So what I'm going to do is I'm going to just map all the things to chromosome 11. So here in the previous step, I just downloaded the chromosome number 11. How many chromosomes does human DNA have? 23. Okay. A bit more elaborate, like what are those 23? How many X, how many Y? And how many of these are XS and YYs? Okay, very good. So, right, so a bit more elaborate, right? So 23 is a blanket answer and 22 plus 1 is the slightly elaborate answer. There are actually 46 in total, 23 pairs of chromosomes. So chromosomes occur in pairs. We are working with the haploid version, which means there is only one copy of chromosome 11 that we're downloading because for all practical reasons, the other copy is exactly similar. Okay. Jayesh, with me? Okay. Okay. Now, why did I ask you to open this RAM column is because we're going to run or create the VWT index. I want you to just observe what happens to the RAM as we do it. And then we'll keep observing this when we are starting to do mapping as well. So the first thing that we're doing is we are installing this tool called BWA, which is Burrows-Wheeler aligner. It uses BWT transform or BW transform that we talked about in the last class to create an index of this chromosome. So index is carrying out one function. It's going to compress the sequence of chromosome 11. It's going to store this in compact format. And then it's going to use that format for doing exact matching of your read data. Okay. I'm creating the index now. So you can see the uptick in RAM a bit. Okay, while this is happening, any questions? Any questions about what we're doing? Any questions about alignment? Any questions about the technology? Should take a couple of minutes. Yeah, what was the multi-QC doing? So multi-QC, we created fast QC reports for individual samples. Now, what you'll have to do if you want to really check the quality of each sample is download this file. So this is R1, then download R2, and keep doing this. So imagine you had like 94 samples. So this is 94 cross 2, which is 188 samples. You'll have to manually see all them one by one. What multi-QC is doing is it's putting them together. And sorry. So I cannot see it here. Okay. What multi-QC is doing is it's pulling all them together in one HTML file. So it's just a tool to bring all the reports about quality in one file, and you should be able to see it here. So R1 is here, R2 is here, and all this information has been collapsed into one file. Good question. I did not elaborate upon it because, yeah, this tool I'm showing you because people use it and they like it and so on. But I mean, it's good. It's good. It's a good tool. But honestly, this step of checking the quality like manually is kind of obsolete. We have good adapter trimming tools now, so they take care of everything. So I don't even check these files. But I think you should check, right? You should always check. But I don't check personally. I don't check quality scores anymore. This was a huge problem around 2009, 10, 11, when this technology was just maturing. I think we are matured enough now to so that we don't have to worry about sequencing technology generally, unless you're doing this in the lab for the first time. That's when you'll do this kind of checks. Okay, let's go back. Have I finished? Okay, so 171 seconds, right? Around 3 minutes. Chromosome 11, one exercise that I did not give you is I should have asked you what is the size of chromosome 11. You can find this out using the commands that we just talked about. So you can do this in Python, you can do this in Bash or awk or any tool that you want to use. Calculate the chromosome size, the size of chromosome 11, so you have a context of how much is the time complexity of creating an index. Okay, next is the interesting part. So I promise this will finish before the class ends. So we're going to now install something called SAM tools. So I had briefly touched upon this format. I'm just going to give you a quick summary. So we have reads, we have aligned them. Now we are going to store, once we do the alignment, we want to store this in a compact format. And SAM or sequence alignment format is the format for storing the alignments. What does it store? It stores the read, the actual read that was sequenced. It stores what is the total number of matches that it sees. So this sequence here is the compressed version of how many matches it has. So 8 matches, 2 insertions, 4 matches, 1 deletion, 3 matches. And then the corresponding reference sequence to which it aligns to. So reference number 7 and position number 30 is where the alignment begins. General idea of a SAM file is clear. So nothing very fancy, right? So there are multiple records, but in most cases, you don't have to worry about all of this. You just get this file and you should just know what exactly it is doing. And it's storing the alignments that we are doing in a compact fashion. Okay. So my SAM tools is done. Keep tracking the memory. Now we are doing the magic step. The magic step is of alignment. So using the BWT transform that we had, so one thing that you can figure out actually what that tool did is you only downloaded chromosome 11.fa, right? But the indexing step created these different files for you. BWT is your BW transform. ANN is internal structure that BWA uses for storing where to exactly begin storing these indexes and so on. So the exact thing does not matter. The only two things that you should care about is BWT and SA. What is SA? BWT is we know BW transform. What is SA? No, there's no sequence alignment yet. This is the index. This is the genome. So there's no sequence alignment yet. So there was some, we started with tries, then we went to trees, and there was one more structure that we talked about. It is that SA. Okay, louder. Suffix and yes, suffix array. Okay, suffix array, just to recall, lexicographical sorting of the suffixes stores where exactly they begin with. Okay, that's as simple. So two data structures very important, BWT and SA. That is what it's going to use internally. Okay, so you would have seen this bump here again as I did alignment. There is a small bump that I see in the memory. Everything is still fitting in my RAM, so all good. We're just using 1 GB extra from what we were previously using, so all good so far. But this step, it's taking all the reads, almost 1 million reads that you have, 1 million read pairs that you have, and trying to find out where exactly do they belong to in chromosome 11. So I have this minus T32 here that you're seeing. I'm using 32 threads. It's sort of an overkill, but why not? And then I'm storing this to a file, which is the SAM file. So whatever BW outputs, it will output on your terminal unless you ask it to output to a file. In this case, I'm saying whatever you see it on the terminal, whatever alignments you're printing it, don't print them on the terminal, print them to a file called the SAM file. Okay, so we'll have to wait. It should be done really, really soon. So very briefly, what it is doing is it loaded that BWT index into memory. It is now reading inputs from the FASTQ file and just figuring out where it does it belong to, right? So it will ideally not load the entire FASTQ file in memory because that doesn't really make sense. So you read it record by record. And the reason you can do this is because you know the format. 4 lines, every 4 lines is one record of the FASTQ file. While this is happening, any other questions? Any other questions where you think I sped through or was not clear, or what you're doing, you have absolutely no idea what's happening? Just let's look at the RAM right now. Look at the RAM. What do you think happened? 2.4 to 6.4 directly. What happened? Copies. Copies what? What platform are you using? So BWT is already done. It's just doing the alignment step. So it's figuring out where should the alignment happen. So when it loads things into memory, it actually doesn't load the entire index because it takes that binary kind of approach. Why load everything? Let's start from the smallest things that we read. If we don't find anything in there, then we load the next chunk and so on. So that's what happened here. For the first few reads, it did not have to actually load everything into memory because whatever was loaded from disk from the first round, that was sufficient. But at some point, the reads were unique enough for which the reference had not been loaded. So it loaded that back and then again, went down because that's not required anymore. So it keeps cycling the memory. This is just a trick that's used in the software for keeping the memory footprint very, very low. So we're almost done, right? 5 more minutes and you'll see what the mutation was. You can actually skip forward. Like if you have gone forward, you can run the rest of the analysis and see what you get in the end. So people who have used tools before, or where is Fennel? Fennel is not there. Okay. Anyone else who has used BWA or alignment or done any kind of this work before? Mostly maybe people from the biology background. Naved? Oh, okay. Amit? No. Nobody has used BWA? You have. Pooja, you have, right? Practice sort of. Okay. Okay. Done. So we have the SAM file. We can look at what alignments happened. So everything got mapped to, or a lot of things mapped to CHR11, chromosome 11. This is the position. This is the sequence of matches. So 24 substitutions, 10, 108 matches, 19 substitutions again. Compact form of storing how many matches and mismatches happen. This is the record number and so on. So you know what the SAM file is. This is what the SAM file looks like. Now we are going to convert it to a binary format, which is why am I going to convert this to a binary format? Some more context. Sorry, some more context. Binary format, which is indexable, which allows me random access. I can access any record on the fly rather than having to iterate through the entire file. Why do I want to do that? Because tomorrow I want to only retrieve sequences that belong to alignments that belong to gene X, wherever that gene X is. What I'll have to do in the current format is iterate through the entire set of records to fetch where exactly gene X alignments are. But I don't want to do that. I want to be smarter. I wanted to store it in a format so that anytime I give you location 1 and location 2, you can tell me what alignments happen in that particular region. You can do a very fast pull out of those sequences. So we convert this into a format, which is called the BAM format. So SAM is sequence alignment. BAM is unsurprisingly a binary alignment. It stores this data in the BGZF format, which is a block compressed format, which allows random access. Random access, I think computer science people will understand. Random access allows you to just get access to a particular chunk of data in constant time. Okay, very, very fast. That's what we want to do. Okay, so none of these steps here are like recipe steps. They are all logical steps. They are there for a reason. In this case, the next case, what I'm doing is I'm sorting that BAM file. So I've created the BAM file here. I'm now just going to sort them based on their location, their start location. So locations on chromosome 11, which happened earlier, should appear first here. You can see that doesn't happen in the SAM format. There is 895 occurring before 892. I want to store this in the sorted format. It makes the retrieval much faster because you can do binary search. Next, I'm going to create an index. So this is BGZip format. The index will tell me, like, give me a very quick lookup table of which records sit in which location. Okay, so that's what I'm doing here. Okay, and then you can see what all files you have. So you have a BAM file, you have an index file. The BAM file is 120 megabytes long. Recall that we started with sequences that were 71 megabytes and 65 megabytes, and we have ended up with 120 megabytes. So most of our data in some sense has aligned. Okay, you could have done this in a much faster fashion rather than writing records step by step. You could have just piped them to each other tool and written that particular thing. This is what this command does. You would get the same output if you were to run this command. This will just run everything in one step. Okay. Okay. In this entire exercise, have we actually forgotten what we started with, right? So fine, alignments are good. What I wanted to see was the sample, which is a mock sample. Does it actually have that mutation or not? Okay, what is that mutation that I'm looking for? A to T. Right? Yeah, A to T, A to T. Okay, so unfortunately, you would want to visualize this entire entire BAM file at once, but you cannot do that because you can do that, but not in Colab because IGV. So IGV is a tool for visualizing alignments. You can run this on desktop as an application. So I've put the link. If you download it and you download your BAM file that you see here on the left half, if you download your BAM file, you should be able to put it up there and visualize the entire sequence. We cannot do that. So I subsampled it. I sample only 5% of the alignments and store it into a new file, which is called sample05.bam. And I'm going to use that for visualization right here in the browser. I don't have to go anywhere. Okay. Okay, so this is where the final magic happens. So what I've loaded here, very briefly, is I've loaded genome HG38. HG38 stands for, okay, I should have given some preview, right? So HG38 stands for the assembly, human genome assembly. Right now, it's at 38 number. These numbers keep on changing. Does not matter for the purpose of this course. You will work, if you're working with human data, you will work with HG38 or GRCH38. Okay. I'll give context wherever it is required for this class. Right now, it's fine. And then I'm going to chromosome 11, loading up a particular location. Okay. And I chose that location because I'm feeding it. I know where this is where the HBB gene is located. I'm passing it the BAM file. Okay. So this is the BAM file. This is what the alignments, this is each individual thing that you see here is one read. Okay. And I told you that there is a sequence. So this is the G, this is the HBB, this is exon 1 of HBB. You can zoom out if you want. See if we do want to do that. Okay, this is the entire HB, not the entire, but this is still exon 1 of HBB genes. Okay. What is this color thing here? Why is it colored? Why is this also colored? Why this colored? Why everything is gray? And then life becomes green somewhere. Like what is going on? Why green? Colors are indicative of bases. A is green, C is blue, T is red, G is yellow. Not slightly, not arbitrary choices. There is some science behind it. Will not go there, but the colors represent the different nucleotides. Okay, you see a T here, but it's A here. Is that the right mutation that we were looking for? Okay, just some more context, right? So the gene starts in the reverse direction here. The protein will be made in this direction, not in this direction. Okay, this is the start of the gene. Okay, let me see if it shows the protein sequence. So it doesn't show the protein sequence here, unfortunately. Okay. So I'll show you one more. So in the final notebook, I'll show you that when you actually go and look at the protein sequences, this is where your valine is. But this is T to A, right? What went wrong? The original mutation that I was interested in was A to T. What went wrong? I also told you what likely went wrong. So this is a sequence. The sequence is fixed. It's based on this idea that everything moves in this direction. Okay. But this gene moves in the other direction. So definitely we have a mutation in this data. Okay. There is one mutation very, very clear to see. Problem is that mutation is likely wrong. Why is it wrong? Well, it's not wrong. The reason it appears to be wrong is because we are reading the reverse complement sequence as the reference here. The gene does not read in this direction. The gene reads in this other direction. So what you're seeing T here is actually an A for that gene. Does that make sense? This C is what? G, and so on. Okay. And this A is what then? T. Okay. Slightly counterintuitive, but not difficult to see, not difficult to understand if you understand what is happening here. The gene reads in this direction. The sequences that you're seeing, they all use this forward direction as the reference. Okay, that's why everything is flipped in this case. So it's slightly unfortunate that I could not show you the A to T, but this is A to T mutation that we have in the data, right? Very, very easy to see. You can browse your alignments. You can go to other locations in the genome. You can browse it. You can do a, you can zoom it out further. I'm going to close this. You can zoom it to whatever level you want. It takes a while for it to load up. But you can sort of do this in an entire genome way, right? So what I've taught you is actually what a lot of companies do, which is, imagine you are sitting at this company, let's call it F-Trend. F-Trend will try to look at, you go to it, it will try to do a panel for you and it will try to understand which genome mutations you have. So it will do exactly the steps that we did. Once you get that alignment data, it will visually verify whether you actually have that mutation or not, was it miscalled or so on. And the way you get confidence is by looking at these sequences vertically align up to each other. So the more number of reads that you see which have A, that is the level of confidence that you get. Okay. So what you're seeing here is exactly what is done in a lot of companies or in a lot of startups, trying to figure out what all mutations you have so that you can be put on one or the other therapeutic regime. Okay. So now you are also a genome data scientist. So like you can figure out which mutations exist in your genome. You can verify it. We have one thing that we have not talked about is the math behind making sure that the thing that you're seeing here, how confident you are. But if you've done the conditional probability, this is not very hard to calculate how much confident you are of seeing a mutation like this. Okay, so the session ends here. But if you're very, very curious, I only analyzed one sample. You should go back, try to look out for other examples. So in this case, you know the mutation. And I also told you that this paper is about a therapy. What therapy was done so that this mutation was fixed? Can you find out that data set and show that this mutation is reversed? That would be like a beautiful, beautiful project in itself. Any questions? Yeah. I see. Okay, so let me come back to you. Anyone who was not able to do this? Or any questions? Okay, so in that case, we'll stop here. We'll start RNA-seq in the next class. My plan was to start it today. We'll start in the next class. Just to give you some preview, we'll talk about the same alignment problem, but they would have